When it comes to developing the right material for the right job, nature is a great engineer that has had a head start of several billion years to create an extremely large number of high-performance, elegant, and complex materials that result in robust structures using as little mass as possible. This is achieved through judicious arrangements of polymeric and inorganic components into delicate hierarchical structures. Natural materials like spider silk, bone, conch shell, tooth enamel, or mollusk shell, among others, are some examples of biological materials with mechanical properties that often surpass man-made artificial materials by orders of magnitude. This fascinates scientists and engineers who over the past 2 decades have been at the forefront of efforts to replicate bio-inspiring structures for fields like biomedicine, energy storage and conversion, or infrastructure. One example of such natural materials is the protein-based polymer, which plays structural and biological roles in many living organisms. Protein-based polymers provide a unique opportunity for the tunable design of polymer systems through genetic engineering or chemical modification. With the advance of characterization and modeling tools, we are now able to understand the complex interplay of mechanisms that act across the different scales of protein-based polymers, from atoms to the continuum. We can develop computational models to study in silico novel fusion protein-based materials. This allows us to predict how they interact with each other or how to combine their different mechanical properties to create materials with targeted functions without the need of numerous experimental iterations to determine fabrication parameters. In this work, we combine the properties of two important proteins in nature, silk and elastin, to create a novel library of proteins termed silk-elastin-like proteins, or SELPs. Elastin proteins display elasticity and stimuli-responsive properties, while silk is one of the strongest natural materials known to man and helps to moderate elastin's responsive features by varying the degree of crystallinity to tune mechanical stiffness. Cells incorporate the unique properties of their composing parts to create an almost supernatural material, generally biocompatible and fully degradable in vivo, which is critical for many biomedical needs. Cells can be used to generate hydrogels that present large swelling ratios as well as reversible changes in optical transparency, mechanical properties, and hydrogel pore size upon exposure to temperature changes, dependent on the silk to elastin ratio. We developed atomistic computational models to explore the effect of different silk to elastin ratios on the performance of the material. The application of computational simulations helps to predict the mechanical properties displayed by cells with different ratios of silk to elastin domains and search for optimal mechanical properties before actually synthesizing the material. Our simulations show that low silk to elastin ratios lead to large swelling ratios in response to temperature changes. By contrast, a higher silk to elastin ratio exhibits no significant thermal response. The reason for this is hidden in the molecular structure of the cell. Too much silk mutes the responsive features of elastin. Once the most promising silk to elastin ratios are identified through multiscale modeling, we can use molecular biology to synthesize them experimentally. We can clone the genetic sequence encoding the most promising SALP configuration into a plasmid that is subsequently transformed into E. coli to induce the expression of SALPs that we can use to manufacture different types of materials, like the hydrogel shown in this video. The resulting materials elegantly validate the models, showing the power of computational modeling for designing novel protein-based materials. The development of these dynamic protein hydrogels based on SELPs paves the way for using these hydrogels as biological structures in tissue regeneration and as vehicles for drug delivery. These results will be useful to guide future library designs with specific targeted properties, leading to new opportunities in biomimetics, biomaterials, and tissue engineering.